Now that we've covered some ways metabolism could function in lieu of enzymes, how did enzymes themselves come about? Going back to the RNP world, a very straightforward explanation would be that it started with a single amino acid cofactor on a ribosome. Later, more peptide cofactors of increasing length would be added, all the while the portion of the biocatalytic unit composed of RNA would decrease until you effectively had an enzyme that we would recognize today. However, the process wasn't necessarily as gradual, and abiotically produced peptide domains could be added to a ribosome. While any one would have nowhere near the specificity for a reaction that modern enzymes do, many random sequences would have rudimentary catalytic functions that could assist the ribosome. While the odds of the same sequence being produced twice by abiotic processes are infinitesimally small, amino acids with similar R groups can perform similar functions in an enzyme. So, even with random sequences being generated, some of them with a similar structure will be produced on a regular basis. Many enzymes utilize B vitamin cofactors, and these are bound to RNA nucleotides, which are thought to be remnants of ancient ribozymes. Others possess magnesium, zinc, and manganese ions that may have once been a cofactor for a ribozyme, where the RNA was completely replaced by peptides. In these enzymes, the most conserved domains are around the active site, implying that amino acid residues played a greater role in abetting catalytic activity in ribozymes than providing structural stability. Many enzymes possess metallic cofactors, and these likely started out as amino acids acting as chelating agents, binding to divalent metal ions and preventing them from breaking down membranes or polymers. Gradually, more amino acid residues could be added to these chelating agents, which would increase and specify what kind of catalytic activity the ion carried out. Finally, adding a mixture of iron 2 and 3 to a cysteine solution will lead to the formation of ferredoxins, where the iron reacts with the sulfur on the cysteine R group. Enzymes that have ferredoxins are bound to them via cysteine residues. While enzymes essentially speed up the rate at which metabolic reactions take place, without an energy source, metabolism will not occur, regardless of whether or not a catalyst is present. Modern biochemistry is driven by adenosine triphosphate, or ATP, more specifically, cleaving the high-energy phosphodiester bond between the second and third phosphorus atoms. Once the energy from the bond is used, an inert orthophosphate molecule is released, and an input of energy is needed to regenerate ATP by reacting the orthophosphate with an adenosine diphosphate, or ADP. Modern organisms react chemicals obtained from the environment or use light to produce the energy needed to regenerate ATP. Apart from requiring energy, modern organisms use a complex system of molecular machinery to synthesize ATP, machinery that would not have been available to the first organisms. Many biochemists have proposed mechanisms by which early life could have used chemical energy in the environment to regenerate ATP. However, the first cells may not have had to regenerate it at all. When life began, a mineral known as schreibersite would have fallen to Earth in the form of metallic meteorites. When submerged in water, it would release reactive phosphorus species, or RPS, such as phosphite, pyrophosphate, and metaphosphate, which life could use. For one, pyrophosphate can be reacted with a biomonomer, releasing an orthophosphate group when the phosphodiester bond is cleaved and forming an organophosphate group. Since phosphates are such a good leaving group, this monomer could react with another, forming a dimer and releasing another orthophosphate, and this mechanism could drive polymerization reactions. Nucleotides with one phosphate attached, or NMPs, can be reacted with a metaphosphate ion to form a diphosphate nucleotide to be used in enzymatic polymerization reactions. Later, another metaphosphate can be reacted with an NDP to form NTPs, which would allow for more rapid polymerization reactions and could be used as an energy currency in a manner similar to ATP. So, instead of using an environmental energy source to replenish phosphodiester bonds, Early organisms could take high-energy phosphorus molecules themselves from the environment and release orthophosphate as metabolic waste. Keep in mind that the time spanning between the first organism and LUCA was covered by the late heavy bombardment. These phosphorylation reactions could be catalyzed by magnesium ions, ribozymes, or enzymes. All the while, complex molecular machinery could have evolved. So far, we've covered the metabolic processes which existed in old. Now, let's shift gears a bit and talk about its molecular biology. One of the first major changes would be the switch from RNA to DNA genomes. The difference between an RNA and DNA nucleotide is that while the second carbon of an RNA nucleotide is bound to a hydrogen atom and an OH group, DNA has two hydrogen atoms on the second carbon. RNA stands for ribonucleic acid, while DNA stands for deoxyribonucleic acid. Some astrobiologists have speculated that RNA and DNA were both present in the first cells and have proposed mechanisms by which DNA could be synthesized abiotically. However, the fact that DNA nucleotides are produced by modifying RNA nucleotides makes this unlikely. It's difficult to comprehend how a ribozyme could modify an RNA nucleotide into a DNA one, since the mechanism used in living systems involves the production of free radicals. This means that if a ribozyme did catalyze this reaction, it either had to have had a cysteine cofactor or used a different mechanism, such as the reverse aldolase reaction. Once DNA nucleotides were produced, a polymerizing ribozyme could use an RNA sequence, such as another ribozyme, as a template to produce a DNA strand. This would be far more stable than an RNA strand because there would be no OH group on the second carbon bumping up against the adjacent nucleotide and could be repeatedly used as a template to produce more ribozymes. Because DNA is so much more stable than RNA, the various oligomers could be joined together or catenated into a single polymer. From the standpoint of evolutionary biology, this would mean that natural selection would now act more on the organism as a whole versus individual oligomers within it, mitigating the deleterious impacts of selfish genes. How did the genetic code by which DNA specifies how proteins are produced come about? The translation of genetic information into proteins includes mRNA, which is an RNA copy of a section of DNA, tRNAs, which bind to a specific amino acid, and the ribosome, which is composed of rRNA and joins the amino acids together into a chain. The tRNAs possess a three-nucleotide sequence known as an anticodon, which is complementary to a three-nucleotide sequence of an mRNA known as a codon, which allows DNA to specify amino acid sequences. So, how could such a system have come about? The tRNAs could have originally been self-amino-acylating ribozymes, or a portion of a ribozyme that an amino-acylating ribozyme bound an amino acid cofactor to. This amino-acylating ribozyme would be the precursor of the enzymes which bind amino acids to modern tRNAs. Then another ribozyme, which carried out aminating reactions, could then bind to the pre-tRNAs and combine the amino acids which they're carrying into a dipeptide. This ribozyme was the progenitor of the small subunit of the ribosome. The pre-tRNA sequences were duplicated, doubling the length of the sequence, forming the distinct L shape, and facilitating the binding of the large subunit of the ribosome. The proto-mRNA initially would have acted as a cofactor, which bound to the anticodon of the pre-tRNA, stabilizing the complex and allowing the formation of peptides with more than two residues. Subsequent duplications and diversification of the tRNAs and the amino-acylating ribozymes would allow the amino-acylating ribozymes to bind a specific amino acid to a particular tRNA sequence, meaning that a tRNA with a certain anticodon will only be bound to a single variety of amino acid. This would in turn allow early mRNAs to evolve correspondingly and specify amino acid sequences in a peptide. When did DNA enter the picture in all this? It's hard to say, but I would argue that in order for the tRNAs, mRNAs, and amino-acylating ribozymes to co-evolve to give rise to a genetic code, the information had to be based in a single genome. So, my tentative answer is that DNA came into the picture by the last two steps at the very latest. Now, let's look at how membranes evolved over time. Initially, the protocell membranes would have been composed mainly of carboxylic acids, but at times, di and triglycerides may have made up a sizable fraction of the lipids, depending on the local geochemistry. The benefit of including these lipids would be a more heterogeneous composition of the membrane, which will in turn increase both the stability and permeability of the membrane. As such, any organism which produced glycerides would be selected for. Adding a charged phosphate group would further increase the stability of the membrane, but decrease its permeability. Bacteria and archaea both have phosphate groups on their membrane lipids, but the enzymes which phosphorylate them are different. This has led to speculation that the two lineages evolved phospholipids convergently. However, the mechanism by which the phosphate group is attached is very similar in the two domains, and it does not resemble any known abiotic mechanism by which phospholipids can be produced. This suggests that the reaction was catalyzed by RNPs in Old and LUCA. Comparative molecular biology has indicated that the membrane-bound proteins which facilitated cross-membrane transport were present when LUCA existed, but were a recent development by this point. This means that if the precursors of LUCA had phospholipids in their cell membrane, they likely would have been mixed with other lipids such as triglycerides and aminolipids to maintain permeability. How did the transmembrane proteins themselves evolve? Proteins embedded in the cell membrane carry out a variety of functions. Some are enzymes, which metabolize exogenous material. Others are signal molecules, and some move molecules across the membrane. Transmembrane proteins remain in the membrane by having a ring of outer alpha helices composed of hydrophobic amino acids. Proteins are synthesized in the cytoplasm via the ribosomes, and if a portion with a large hydrophobic region was synthesized, said hydrophobic region would prevent it from floating off into the surrounding liquid, keeping it bound to the ribosome. In order to get from the ribosome to the membrane, a translocon is required. Translocons supposedly would not have been needed before transmembrane proteins, which leads to another chicken and egg problem. One manner in which transmembrane proteins are hypothesized to have evolved is that the first alpha helices were amphiphilic. These would have been composed of peptides with two alpha helices, and the amino acids on one side of each helix would be hydrophobic and the other hydrophilic. In the cytoplasm, the helices would be folded with the hydrophobic sides adjacent to each other, while in the membrane, they would flip with the hydrophilic side adjacent. Many of these peptides could then condense to form a quaternary structure resembling a pore in the membrane. This mechanism is known as the inside-out hypothesis, and it is used by many bacterial toxins to this day. As the integral membrane alpha helices became more hydrophobic, the translocons could co-evolve with them.